Hello guys, my name is Dr. Varun and you are watching Chlorophyll classes only on YouTube. Well, here we're gonna be debunking your biology concepts problem and we will try to make you fall in love with biology. Well, today we're gonna be studying about the 12th class, and what we're gonna be studying? We're gonna be studying about the molecular basis of inheritance chapter, and today the topic is transcription. Well, this is the most topic which are being always a scary part of the student's life. Let's try to make it easier for you. Starting it up, what is transcription? If you remember about the first and second parts of the chapter, if you've not gone through it, go back and see it. So in that chapter topics, what we have studied, we studied that since the what basically happens, DNA contains all the information, means whatever I body, if I'm doing like this is being what it is being DNA which is able to make me do so, right? What I'm thinking, but how it is making me do anything in my body, any shape, any color, anything in my body, how it is able to do it directly. DNA of each cell is not working. Okay, it's not like that each DNA is like okay, we are double stranded, let's do this work. No, they make something as a their person who will do it. So for example, if you go to any startup, in startup what happened, there is a founder who has a vision. So founder is what? DNA. Okay, that founder will do what? It will hire people. So what will be the people? There will be the people who will work for them. For example, there will be interns, there will be managers, there will be CEOs, all of them work for them. Okay, now whatever that people that are there, this people process of hiring, in this we're gonna be calling it transcription. Able to understood? Let's give another example. In school, what happened, beta? In school, you know that your principal has a vision. So principal will hire a lot of teachers for it. So the teachers would be what? Teachers would be the middle person, and on you we will work, and the results that we will get, like for example, you will get 100 marks in biology, 100 in chemistry, that same thing. That is what? That is the effect. That's the final thing that we wanted to happen. But for that to happen, you need a teacher who can help you out teaching that thing. Without that, nothing happen. Even the AI is what? Acting like a teacher, right? So this thing we will remember it. So fact, we understood what we're going to be doing. We understood and why we are doing it, because DNA directly won't do the thing. It needs what? A middleman. So middleman would be what? This process product that will come up. That product process, like the product of this process would be what? Formation of RNA by the DNA. So RNA is our teacher or are the hirings from the startup. Clear? So it is what? It is the transcription is the process of formation or synthesis of RNA over DNA. Now, before to go into it, let's just talk the basic thing. Now, DNA is very big. There will be, for example, if there is one part of DNA which is responsible for what? For my eye color. Okay, so what happened? During the growth time, when my eye color has to be made, what will happen? DNA of those cells which requires, they will make what? They will make the RNA. RNA will make the proteins which will give the colors to my eye. This basic thing understood. So means that if DNA is making RNA, we should know that what are the basic things, few things we have to remember. Now, if you remember the replication, we used to say what? That if this is the template strand, on that what will happen? DNA will form. One new strand would be continuous synthesis, other would be what? Discontinuous synthesis. Now, if you remember a little bit about it, what happened? You know that if this is the 2 strand, 5' to 3', this is for the replication I am talking about. Okay. This is antiporal. What will happen if the new strand is forming? One of this would be here, one would be here. Okay. And on this, what will happen? New strand will form. So the new synthesis strand polarity will always be like this. Okay, here also new strand that is forming, its polarity would always be like this. So if someone will ask like how the new strand is forming, it is forming 5' to 3'. This is the polarity of the new strand. Older strand polarity would be what? See from here what it is going. From this side it is going towards this side, means 3' to 5'. This thing you have to remember. Here also the primer where it will go and attached on the DNA, it will again attach on the 3' only. The primer is not able to attach on the 5 dash even in case of transcription process also. So one thing is similar. If it gonna be adding on the 3 dash proximity or the closest to the 3 dash, it means what? The new part that will form its polarity would be what? 5 dash to 3 dash. So the same way your DNA has polarity, same is the RNA. So till now we haven't talked about RNA. We will study about RNA. So RNA is exactly similar like your DNA only. The difference would be what? The difference of the RNA is that the RNA has uracil in place of thymine. We are talking about a strand way. Okay, RNA is single stranded only if it is working as a in our body. Okay, in our body it's not double stranded. Why not double stranded? We will talk about these facts also for the RNA. Okay, so this thing we understood. Okay, what else changed? Instead of the deoxyribose sugar, there is ribose sugar. Okay, that's the only difference it has. Okay, so it can easily act like what? It acts like a substitute. Okay, for the new strand. So again it will be coding the same thing. Okay, certain coding related questions will be there. We will discuss it. Don't worry, we are not gonna leave this chapters haphazard. This is the first layer of this chapter, to be very honest, on the YouTube. So now if we need certain things for it, we need to understand this polarity concepts. So what would be the polarity concept? Again, I'm going to be making a green color DNA. Okay, on that the RNA will be forming up. So here, to make it easier, I am keeping it what? I am keeping it stretched. Okay, so that you can see that what's happening. So what happened basically, if this is your DNA part, so this part I am stretching it. Okay, similarly, this would be what? DNA part. This is a stretch portion so that you can see it. Now this DNA will have what? It has polarity 5 dash, 3 dash, 3 dash, 5 dash. Okay, it has what? Its nucleotide sequences present over here. Throughout it will be present, right? And you know that if the DNA is there, both the strand would be what? Opposite. And you know that there will be pairing of A to D and C to G or vice versa. So this, okay, I try to make it. Okay, this is your what? DNA. This is your DNA molecule. Now the whole of the DNA will not make RNA, only the portion which is doing that. That's the eye color. For example, this eye color portion will act. Okay, so one fact what we will remember, only certain portion of DNA will make one type of RNA for particular function. Means what? Particular teacher for the particular subject, like that. Okay, now over here what will happen? I told you what? RNA will, like anything, it has to start the process. We need a foundation stone. So here also a foundation stone will come, and where it will act? It will act either at the 3 dash of it, but which one? Okay, so basically that depends upon the upstream and the downstream thing. So if I am taking this green color one, darker green color one, so the 3 dash is proximal to this area, right? This part is 3 dash of it, whereas this is what? This is closer to the 5 dash of it. So it won't come, it comes like this, and it present upstream, above. So the DNA will open up little bit over that, and it will go and attached over there. It will keep on moving like this, and then it will go downstream. Downstream means what? It will comes down. So here what will happen? This red color thing is what? Where the priming will occur. So this is called as what? Promoter site. Okay, so what we're going to be learning? We're going to be learning now the facts about the apparatus. Okay, so it is what? Apparatus of mechanism. So this thing is what? This red color thing. This red color thing is what? It is the promoter site. So where it will be present? It will be present upstream at 3 dash end. Okay, now the next thing is upside down. It could be done. Okay, if it would be other way around, means if this would be acting, so 3 dash is on that side, this will come here, but present towards upstream. Okay, so this one fact we understood. Okay, then comes what? Then comes our this in between, this whole thing that is present. What this? These are the nucleotides which are present over here. These has to be only added up now when this promoter primer will move. Okay, whatever it is, we will learn it. When it will move, it will keep on adding new nucleotides behind it and making a complementary strand to it, right? So this thing is called as what? This thing is called as what? These are the structural genes. So these are what? Structural genes. And once it reaches to the end point where it has to stop, this thing comes down. Okay, I think this would be a better one. This thing would comes down. So this is present on what? Closer proximity to 5'. So this is what? This is the terminator site, termination site, which is present downstream at 5' end. Here it would be present on this side, downstream. Okay, if we are taking it like that. This thing we understood. Now the new genes that will be forming, it will always be in the 5' to 3'. Okay, so means transcription is occurring what? It will be forming 5' to 3' thing. Okay, because its complementary thing will form. Okay, so here we will remember the fact, other thing than this, that would be what? That RNA transcript in 5' to 3' and only. Okay, so this is the site 3' end where what? Where primer will attach to initiate the process. Okay, what it is, how it looks like, we will learn it about the primer. Moving on to the next thing, transcription is occurring 5' to 3' only, means, and it is what? And RNA formed is complementary to template strand. So here with a blue color, I'm going to be showing you what is a template strand. Since everything is occurring on darker green color, so this darker green color would be what? Our template strand. So this is our what? This is our template strand. It is also called as anti-sense strand. Why anti-sense strand? Because it is opposite to making the sense of the your RNA. Means RNA will have A, it will have what? T, DNA strand, opposite. So it is anti-sense, opposite to it. So this strand is also called as anti-sense strand. Whereas if it is opposite, means this strand sequence will be also opposite to this also. Means this thing and the new strand that is formed, mRNA, or the RNA that is being formed, it is what? It is will be similar. So can I say that the opposite strand of the DNA, this lighter green color, that would be what? That would be the coding strand, because it will have same thing like the RNA that is being formed. I will explain you, don't worry. And if it is the same thing, means it is also what? It is making sense. So this is the sense strand, light green one. These are certain terms you have to remember. Here I am writing it again. What we're going to be writing? The RNA transcript or formed on template whose polarity is 3' to 5'. First thing, since the RNA will be complementary to this DNA strand this DNA strand, that would be better. This DNA strand is called antisense or template strand. Now, whereas the non-template strand of DNA with polarity 5' to 3', this is we talking about the DNA. Okay, polarity with 5' to 3' has similar nucleotide sequence, except as what? Uracil and thymine position. Okay, because thymine is not present in the RNA. Nucleotide sequence, except, in bracket you can write except at that. Except at thymine position is called sense strand or coding strand. Understood? Very important, very important for a lot of questions. Let's try to see that with an example also. Over here, the questions that can come. We are doing the basic things only. We are not going very deeper into it. Don't worry. So I'm making a strand for you. Okay. Keeping up. Okay, now this is the strand, right? And its opposite would be what? If someone will ask you what would be the what would be the nucleotide sequence on this DNA? Okay, this is what? This is DNA. How I know it is a DNA? Because it has what? 2 strands present plus T is coming. So T will only be present, thymine will only be present in the DNA. Okay, that's a difference also. Okay, now opposite strand, if I have to write it up, what I will write? I will write this thing. A will be T, T this, C, because I know that A will make bond with T, C will make bond with G. We know this fact from the Chagas rules and the Watson and Crick model. Okay, this would be, C would be this, T, A, C, G, C, T, G, C, T, A. Okay. Now what I did, I took this link. Now I am giving up the certain terminologies, further terminologies. Now this is what? This is my gonna be the template strand. Template strand. Okay. So if this is the template strand, make RNA on it. So what would be the RNA sequence? This is the question. If this is the template strand given to you. So first thing, what you will write? You will write that whatever be the sequence, there will be what? There will be, it will be forming 5' to 3'. Always, if you are given this, you have to write the sequence in the 5' to 3', because that is how it will be forming, 5' to 3'. Don't write 3' to 5'. You have to see this also in the paper. So now when you would be seeing that, what will happen? This T, whatever it is, okay, this T that is present, it will correspond to this area. This will correspond to this area. This will correspond to this area. Can I write it? I'm writing with the pink color only. So this T will becomes what? A, because what is what? We know that whatever will be transcript would be what? Complementary, in this way only. It's only that in place of A, it will be U. That's the only difference. Okay, so this T was there, the T opposite will be A. A opposite will be what? U. Because I told you that in RNA, there will not be T. So where A corresponds to T in the DNA, here replication, but here it will, when that comes to transcription, it will correspond to U. So it will bring the U from the cellular area and form this. G will correspond to C. This will correspond to C. Then T will correspond to A will correspond to U, C will correspond to G, C, G, A. This G is there, so it will be C, G, A, U, U, and U. So this is what new RNA sequence. So this is what the RNA sequence. If you see this sequence and if you see this strand, it's same except at the place of T. So if you see that A, T is changed by U, then C, A, T changed with U. Only this is the change. Then G, C, G, A, C, G, A. T will be changed with the U. Which means what? These two strands. What beta? These two strands. They are what? They are same in sequence. And if they are same in sequence, can I say, okay, can I say that the sequence of 5' to 3' of the DNA is same, means it is the same code also? So if I know the coding sequence and they will ask me in paper that write the RNA sequence, I have to exactly write the same thing, just what? Change the position with T with U in the RNA. Easy. Fastest question to solve. So in paper, if they are giving you coding sequence, okay, so that's why this is called as what? Coding strand. Then you will just write the same thing in the RNA. Moving on, and this is the complementary ones. This is what? This is complementary. This is the most important question that you have to remember to approach. If we understand this, let's start the process. So the process start with what? It will be started by what? By the help of a primer. So here, who will play that? That would be done by RNA polymerase. So process will be started by RNA polymerase. Now in case of prokaryotes, there is only one RNA polymerase. But in case of eukaryotes, there are 3 different polymerases. Now the major thing is that the RNA in the prokaryotes is making different types of, RNA polymerase in eukaryotes makes the different types of RNAs. So we have more types of RNA. Here I am giving you just an example. We have rRNA, we have tRNA, we have sRNA, and many more than this is also present, but that's name only we have to remember. We don't have to see them in much detail, not very much detail anything, but just the name of that. But these 3 we will be studying little bit more about it. Clear? So what I told you, that for this process to start, there is a requirement of a RNA polymerase enzyme. This RNA polymerase enzymes would be what? It would be one present. It will be one only present in case of prokaryotes. It will be multiple. Okay, 3 different types in the eukaryotes. So we're going to be learning about the enzyme required. Same way like over there, here also we're going to be studying about it. About this enzyme only we're going to have to study, not much. Of course, there are more than this is required, but we're going to be studying this only. Okay, enzymes. So what is the enzyme? The enzyme is the RNA polymerase. So same that DNA polymerase used to what? It keeps on completing the process. This also do it. Okay, so RNA polymerase is what? In prokaryotes, only one type. For all the RNA that will be forming inside the prokaryotes, only one type is present, which forms all the type of RNA. Okay. In eukaryotes, three different types are present, and we have to remember that. What are they? They are what? RNA polymerase 1, RNA polymerase, and this is the important one because given in your NCERT also. Okay, then RNA polymerase 3. What we have to remember? What they are making up. Okay, so RNA polymerase 1 is making what? It is making your 18S rRNA. This S is what? Unit of it. Okay, 18S rRNA, 20S rRNA, 5.8S rRNA. So this is RN, one type of RNA. Then we have polymerase 2 that will be making mRNA and hnRNA. Okay, whereas polymerase 3 will be making tRNA and 5S rRNA. Okay, in cell chapter we will be learning about these things again. So over there you will understand much more about this thing. So it's the linking of the two chapters, and from this the question also come recently. Okay, not very far off. It's not out of syllabus. It's very much in syllabus when we're going to be studying that thing. So till now what we studied? We studied that for this there is an apparatus, three things required: promoter site, structural genes, which is also called a cistron, and the termination. It also requirements of what? It's required an enzyme, RNA polymerase. Now how does RNA polymerase look like? Now this RNA polymerase is made up of peptide of chains. There are different types of peptide of chains which comes together and form it. Okay, so we're going to be studying about this RNA polymerase a little bit more. Okay, so what it is? RNA polymerase is made up of different types of polypeptide chains, that is alpha 1, beta 1. Along with that there is a sigma factor present. Okay, now before that sigma factor is present, what it is? These four will make what? A core enzyme. Okay, these four makes core enzyme, which will do what? This will identify also, along with that do the structural genes also. So it will do what? It has two function. Number one function is what? First of all we have to remember what? It slides over DNA template, acting like a primer. Okay, and adds nucleotides to RNA. Along with that, here what happened? If you have to identify, along it can't ID it combines with sigma factor. So there is what? One sigma factor, and helps and recognize the site from where we have to start it, and recognize the site to initiate the process. Now when we are saying it's glide over there we never mentioned sigma factor, because sigma factor is removed from it. Because if sigma factor is not removed, then the smooth gliding will not occur. Means that if this is the DNA, on DNA it is making the thing, it won't be able to move. It will move like jerk. So that smooth movement and fast process to take place, What it happens after identifying these 4 peptide chains, which is called as what? It is called as holoenzyme. So it is called as core enzyme. Okay, so this core enzyme, it will just throw the sigma factor out so that it can work. Okay, so initiate the process. So core enzyme plus sigma factor together, they will be called as what? They will be called holoenzyme. Okay, here sigma factor is removed as it creates, so it is what? Use and throw, which lot of your friends do with you. If it is so, let me know in the comment section. I don't know what I'm asking to write in the comment section even. As it creates obstacle in smooth movement or gliding of core enzyme during chain formation or elongation. So if I have to make a rough diagram of it, how it will look like? So it will be like this. So there is alpha, alpha 1, beta, beta 1. They are forming what? They are forming the core enzyme. Whereas there will be what? There is the coming up of sigma factor. Okay, and when, okay, this don't look good. Then comes the sigma factor. Now as this sigma factor comes, this whole structure, okay, better color uses, this whole thing will be called as what? This will be called as holoenzyme once the sigma factor has been added to it. Okay, so we understood that termination, like there will be where the site it will start and there will be ending. During ending beta, what happened? The sigma factor is not present, but the ending to happen, this is what termination point. At termination point, a rho factor comes up. Okay, so that we will learn later on, okay, about it. Okay, so end process, so we will remember that during termination where it has to stop. So at the terminator end, at the termination point, this core enzyme joins with rho factor and terminate or stops the process. Terminate the process. Okay, we are clear with this terminologies and everything. Let's start with the mechanism. Okay, so mechanism would be divided into two. First is the basic, the RNA forming, and then later on what? A post-transcriptional process, means where we are, what we're doing, we are enhancing its capabilities and the products. Okay, so we're going to be starting with the mechanism. First is what? Process of formation first. Process of formation of RNA. Okay, and second is post transcriptional process to make it more processed and mature enough, stable enough. Okay, starting with the first process. Okay, so that would be what? First step would be recognition and initiation of the transcription. Okay, at the promoter site. So first is what? Recognition and initiation of transcription at promoter site. Okay, this is Again, we're gonna be writing it up. Recognition and initiation of transcription at promoter site. Okay, so first of all, RNA polymerase in prokaryotes will recognize a site. We have to remember about how the site. So, okay, so in prokaryotes and in eukaryotes, okay, we're going to be writing it with this different color. In eukaryotes, so we have to remember what we have to recognize in each of them. So in prokaryotes, what happened? The RNA polymerase, which is only single type, it will identify a Pribno box. Pribno box is what? It is a special sequence of 6 nitrogenous boxes lying after the 10 nitrogenous boxes. Okay, 10 nitrogenous bases, which is very much before the promoter site. So means if the promoter site is present, there will be 10 nucleotides, and before it, this site would be present. Okay, so means what? That if I am taking this as the sequence, imagine that only one strand I am taking. Okay, this strand is what? This only. Okay, my template strand. On this is my sequence. So what happened? There will be 10. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And over here, there will be what? 6 different sequences would be present. 1, 2, 3, 4, 5, 6. So I have made it purposefully like this. So what would be present? There would be present of this as what? This is the Pribno box. Okay, this Pribno box is lying 10 nucleotides prior. So if you count this is what? 10 nitrogenous base, or 10 nucleotides would be better term. 10 nucleotides, and this is what? This is your promoter site. This is the promoter site. Understood? So what I said, there is a Pribno box, which is what? Special sequence of 6 nucleotide base, which is present in front of 10 nucleotide base. After that, only promoter site would be present in prokaryotes. So it is what? RNA polymerase will identify promoter site with help of Pribno box, that is special sequence of 6 nitrogenous bases. 6 nitrogenous bases. What are they? TATAAA, or it is TATAAT, which is lying 10 nitrogenous or nucleotide base, nitrogenous bases ahead promoter site. I hope this is understood. I have also write it up. Moving on to the next one, in eukaryotes, there is a TAT box present. Okay, and it is also called as Hogness box. Okay, and it is responsible for identifying the promoter site, and this sequence is present 20 nucleotides before the promoter site. Okay, so same thing over here also. Shall I have to make it this time, or we just write it? Okay, I will make it, no issue. This is also what? This is your sequence. Okay, I am taking that. This is my, so this is what? This is the promoter site. Promoter site, and we have to wait for 20 nucleotide sequences. Okay, so 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. So this is what? 20 nucleotides. which is present, 20 nucleotides which is present, and in front of it there will be what? There will be a special sequence of 6-7 nucleotides. Okay, so 1 2 3 4 5 6 7. So this is what? This is your Tart box. It is also called as Hogness box. Clear? So it is what? It is the RNA polymerase, whatever the type of it is, polymerase will identify promoter site with help of Tart or Hogness box, that is sequence of TAT lying 20 nitrogenous base ahead, like 20 nitrogenous base ahead of promoter site, This is the difference between prokaryotes and eukaryotes identification sites. Now you know that RNA polymerase helps with the help of sigma factor recognize this and initiate the transcription process. So here what will happen? The DNA sequence at this area will little bit unwind. Okay, so unwind duplex will be done, and this RNA polymerase itself will do, and transcriptions will occur at 5' to 3' direction, which we have already studied. We're gonna be just writing it up. So what will happen? The RNA polymerase with help of sigma factor identify and initiate the transcription. DNA duplex is unwinded at the location. RNA transcription occurs at 5' to 3' direction. That's it. It's about the initiation. Now chain elongation is exactly similar like the DNA replication. So what happened? It will act like dual purpose. Okay, nucleotides. It will give what? Triphosphatic form of the nucleotides, breaks down, forming the monophosphatic form, releasing energy, which we'll use in the process. Along with that, it will act like a substrate. So it will be exactly same. So the next thing is second step, that is chain elongation. In chain elongation form, what will happen? That nucleoplasm, very important thing, where it is occurring in the nucleus only. This is very important, which I forgot to write every year. Occur in nucleus. Okay, this transcription. Now nucleoplasm contains ribonucleotides, which acts as dual, or which serves dual purpose, not acts, which serve as dual purpose of energy release as well as act as substrate for the RNA strand. So we know that similar like what DNA happens. Okay, similar like DNA replication. That's it. Then the chain termination means as it reaches to the end point, because rest every steps we have already discussed and understood. As it reaches to the end point, what happened? This core enzymes was like, as this reaches a rho factor comes and it helps in the removal of the chain from it. Present at what? Present at the downstream, 5' downstream of the template strand. So the third is chain termination. Rho factor associate at termination point and stops, but where? Termination point at 5' end downstream of template strand and stops the process. Normally the new forming things, okay, is what that is called as hRNA, okay, and the newly formed RNA is termed as hnRNA. That is what? Heterogeneous nuclear DNA. Heterogeneous nuclear DNA. Sorry, RNA. Now, what is this? This heterogeneous RNA is basically what? It is a unprocessed RNA. Means the process is not completed. First step is completed. Your RNA fragment has been made. But this RNA fragment is not what we say that processed or mature. In this form, it is of no use to in the eukaryotes. But in prokaryotes, it is. Okay. So here we have to understand certain facts. In prokaryotes, okay, first of all, before going to it, let's see that this RNA contains structural nucleotides called as cistron. Now the problem is what? These structural genes which are present over here, they will be what? In prokaryotes, no issue, because they will be doing what? The fragments, part of it will making different proteins to it, because next step is what? Next step is that this RNA which is being formed, it will make the proteins, if you remember the reverse transcription. Now here what happened in the prokaryotes, it's not problem, because single RNA will make multiple proteins, and it can only make it because this is the one portion, it should be obstructed, then the next portion making something. But this obstructed part which is present over there, it's code for nothing. Means these cistrons, certain part of this cistron is what? Working, certain not. So here we're going to be learning about it. So cistron has what? Two different types of genes present. One is what? It has exons present, and other is what? Introns present. Okay. Now exons actually codes for protein. They do not code for protein. So why they are even present? Because it is what? They are being formed in the process. How, if they are not important, then why don't leave it? Why the cell don't stop making this thing? So actually to do that, the cell goes to little higher level of organization. and it forms a eukaryotic cell where these introns we have to remove it. So means the step has been increased, and that step is what? Post transcriptional step. Now here we understand that the cistrons are two types, exon and introns, and we don't want the introns in the eukaryotes, but in prokaryotes it is present. Why? Now the question is this only. So we're going to be understanding that. So in prokaryotes, polycistronic RNA is present, which means what? That single RNA which has multiple different types of cistrons present. Poly means many cistrons present, which is coding for something. This cistron will code for certain proteins. So in prokaryotes, single RNA will form multiple different types of proteins, because smaller primitive one, they want to do more work within the same small processes. So they need this kind of this. But later on in the eukaryotes, what happens? Single RNA will make only one type of proteins, not any other type of protein. Okay, so this happens what? As in them, single RNA forms many or different types of protein. Hence, introns acts as stoppage point for one protein chain. Means it is an obstruction protein chain. So for example, if this is our RNA that is being formed, so this is what? This is 5-2, 3- RNA. Here, there are two different paths present to it. So, this purple color thing is representing what? It is representing the introns. And this yellow color thing is representing what? Exons. So here what happened? This exon part only gonna be making the protein. So if it is what? It is a polycistronic RNA. Polycistronic RNA. What will happen? That this part, okay, wait. This part will make one protein chain. This part will make second protein chain, and this part will make third protein chain, and each protein chain will be doing what? Different function. So this has different function, it has different function, and this has different function. Okay, this happened in the prokaryotes, but this type of polycistronic is not present, because in eukaryotic monocistronic RNA is present. Means that one RNA will make only one protein. So what happened? This and this part has to be taken out, and this thing is called as splicing. And later on, after splicing this part, we have to add this thing. So this thing is called as what? Editing. So we are cutting and editing back again that stuff. So another thing that we have to remember, but in eukaryotes, monocistronic RNA present. So in them, introns, which code for nothing, are removed and exons are joint. This process is called splicing and editing, and that is a part of what? That is a part of your post-transcriptional process. So monocystronic RNA will make what? Make single protein only, not multiple protein. Okay, which is very specific. So now that comes that in eukaryote the step is increased. So we have what? Next topic is post transcriptional modification or changes. So here we will be remembering what? We will be remembering that the fact that is only in eukaryotes, and the reason why only in the eukaryotes is being told in the last. Okay, because they are monocystronic. Okay, so here what will happen? There will be two steps to it. So number one step, that is splicing and editing. So what is splicing and editing? In splicing and editing, the introns, non-coding sequence of snRNA is removed by cutting it with the enzymes. Okay, and exons expressing sequence or coding expressing sequence or coding sequence for protein, okay, are joined back by ligase enzyme, joined back to form processed RNA, for example mRNA. Okay, so what will happen if we see? So this is your 5' to 3'. It has this part 1, 2, 3. Okay, so 3 introns are there. So these are what? These are introns. This is intron. This is intron. So what will happen? We will be forming a loop out of it. They will join together, cut, and rest the parts joined up. So let's just see that. So these will be looped out. So here what will happen? This part, this part, this part cut up. So, and what we are left out with? We will be left out with a processed RNA which is joined together. This is 5' to 3', and these loops part are removed. Removed. So this removed is called as splicing, joining back is called as editing. Okay, so this thing is joined. Okay, so this is what? 3 part. Okay, so this thing is called as what? This is the step-by-step process. This is called as mRNA. It is what? Mature or processed RNA. Now since the process is not correct, because this RNA which is just formed, it's very unstable. If it will be present like this, it will be most probably degenerate and it's not able to form the proteins. So we have to make it little stable. Okay, so next part is this RNA is unstable. So to make it stable, the second process is being done. That is what? Capping and tailing. Capping and tailing. In capping and tailing, what will happen? We will add certain part on the top of, like, the head part of it, and at the tail part certain parts of it. So in capping, what will happen? At 5' end, that is the head part of it, we add 7-methyl guanosine, and at tailing part at 3' end, we add 200 to 300 adenine nitrogenous bases. So it is also called as adenylated tail. Adenylated tail. And this makes it what? This makes this whole substance a more stable. Now it is being transported from the nucleus towards the outside, okay, for the protein synthesis process. So from the nucleus it goes out. That's it. Now this splicing and editing, this feature, this tells us that it has been like this RNA comes from prokaryotes to the eukaryotes. It's a kind of a feature which tells about the evolutionary history of the molecule's development, which leads to formation of processed RNA, or the organisms which are more better. Now let's comes when the RNA is being formed. There are different types of RNA present to us. Let's just quickly look into it and move on. So the next topic is RNA. Same way DNA, RNA is also an important topic for us to remember. So first thing is that mostly it is single-stranded, but it is present in the double-stranded if it is genetic material, and that is in some of the viruses. Next thing is that why not 2 RNA forms? Because if the 2 RNA will form, what will happen? The both RNA that will be forming from a single strand, it will be what? Complementary to each other. So what will happen? They will just make similar. Like first thing, what will happen? If they are complementary thing will make what? Bonds, and form a double bonded structure. And RNA can't function if it is in the double bond form. First thing. Second thing is that if 2 different RNA will form, and if they didn't came closer and make the bond, they will give 2 different characteristics which will be opposite. Like for example, if you remember from the genetics, in genetics we studied what? We studied that if there is one blue color gene and one like for example this, if this is the blue color gene and this is the brown color gene, we say that only one will show it effect. Why? Because only one RNA will form. Now you understood why we say that only one, because either this gene RNA or this gene RNA will form, not both. Both the genes will not form. So if this will form, this becomes what? The dominant one, or the character which is showing up. Now the next thing that we have to remember, if both will form, if this is form, this is form, means one eye will be blue, one eye will be brown, or both the eyes will be mix and match. That never happens. So understood what are the 2 things? This we're gonna be writing it up. Okay, so the RNA is what? With terms of RNA, it is mostly single-stranded if non-genetic. In genetic it can be. Now the thing is why? Because first part, since the genes might be contrasting trait, and if 2 contrasting RNA will form, it will make contrasting proteins, or expressed, or better way is like expressed contrasting traits which is not possible because only one trait will be visible, okay, usually. And second thing is what, that inside cell the contrasting RNA will be complementary to each other, and it might forms a double-stranded structure. A double-stranded RNA could not express, because it can't make protein, it can't express it also, could not express the characteristic. So means I hired a teacher and the teacher is not able to do the subject. That's the thing. Now this nucleotides, if I have to ask the structure of it, structural unit of RNA. So it is what? It is also a nucleotide only. So it is what? Nucleotide. The nucleotide will be made up of what? It will be made up of nucleoside and phosphate. The nucleoside will have what? It will have a sugar and it will have a nitrogenous base. Sugar would be what? It will be ribose sugar. So at the 2' end, there won't be any OH removal. It will be OH present. And here, there will be what? There will be purines and pyrimidines. There will be what? Purines, pyrimidine present. So purine would be what? It will be adenine and guanine. Pyrimidine would be what? Cytosine and uracil. Instead of thymine, uracil is present. Okay, deoxyribose sugar is present. Okay, now the splicing of RNA has shown that the feature that RNA was forming first, then the DNA was being formed. Okay, and RNA could be at a genetic material also. It could be act like a non-genetic material. Okay, so next thing is that RNA can act as genetic material in what? In the viruses. Now why it is not acting like in the other organisms? Because it's a low chemical stability. We have already discussed about it. So act as genetic material in certain viruses only. In certain viruses only, and over there it could be single stranded or the double stranded. Example TMV, tobacco mosaic virus of the 11th class, chapter number 2, which we have to, which do what? Potato splinter disease. Okay, potato splinter, that is viral. Tobacco mosaic virus will be doing what? Mosaic formation, leaf mosaic formation in case of tobacco. Leaf curling will be there, all those things. And AIDS virus. So these are what? This is the, AIDS virus is HIV virus. Okay, so this will be what? Acting like a RNA as a genetic material. Whereas RNA, mostly in eukaryotes, acts as messenger of the process. Okay, messenger of the process means flow of genetic material is being taken care by that. Means what? It has a purpose of formation of lot of things. Okay, and helps in translation process, which is the next process, formation of proteins from the RNA. And this non-genetic, and this is what? It is a non-genetic nature. This non-genetic RNA has different types. So what are these? mRNA, sRNA, sorry, mRNA, tRNA, and rRNA. Okay, so there are what? 3 different types of it. What are they? We have mRNA, which is called as messenger RNA. Okay, we have rRNA, which is called as ribosomal RNA, present inside the ribosome. And the third one is tRNA, called as transfer RNA, which help in transferring of the amino acids during the time of the protein synthesis. Okay, so we're going to be studying. So it consists of what? 10 to 15% of total RNA. Total RNA in cell. Next point is that nitrogenous form bases, they are present as a sequence on the RNA called as codons in the triplets form. So nitrogenous sequence on mRNA forms codon, that is triplet, means 3 of them, like A, it forms one codon. Like this we used to remember it. Okay, now the sequence of mRNA determine the sequence of amino acid of the protein chain. Okay, now here the sequence of codon determines what it is, we will run it, determines the sequence of amino acid in protein chain, which is also written as polypeptide. Protein chains. Okay, what it is, we will learn it right now. Don't stress. Okay, now the rRNA is called as what? It is called as ribosomal RNA. Okay, this is the messenger RNA. Messenger RNA. This is ribosomal RNA. Ribosomal rRNA. And this is transfer rRNA. Now this ribosomal RNA, it is what? It is the most abundant. It is the most abundant. Okay, it is the most abundant. Around 80% of all rRNA in a cell. Okay, forms a major constituent of ribosomes and is present inside what? In the nucleolus also. So present in ribosome and nucleolus. What is nucleolus? Nucleolus is the structure present inside the nucleus where only the ribosomes will be forming. This will also be coming like that. So means certain close relationship might be there between them. And the last one, tRNA. Now this tRNA is the least present, least abundant. That is around 5 to 10% of cell total RNA. But it has a responsibility of carrying the amino acid during the time of translation. So it carries, that was the transfer, carries the amino acid. What it is, we will learn it. Here just I'm writing the fact so that later on when you come back, you can remember it. Carries the amino acid during translation. Now this tRNA is very important for us and we're going to be learning about it, tRNA. Okay, and there are different types of tRNA basis of which amino acid it will bring. So means tRNA is very loyal to the amino acid. Just remember that. Okay, so here we're going to be studying about its structure. Now if you see the 2D structure, it will look like L-shaped. But if you look the 3D structure, it will be what? It's a clover leaf shaped structure. And this clover leaf shaped structure we have to remember. So it is what? Structure of tRNA. Now let's see. So since a RNA will have what? 5' to 3' end. Okay. So here the 5' end. Then it goes like this and it make loop like this. Further it goes, it make loop like this. And over here it makes a short arm, small arm, then make a bigger arm like this, and it goes like this. basically what happened, why this structures comes up, if someone will ask, like this structure comes up because of the what it's a like a clover leaf like structure. And the reason why this structure came up, because this there is what infolding of the tRNA. Means, for example, if there is one wire and it fold up itself, so we call it what, upon itself it folded up and made what, certain paired kind of a structure. Okay, how we will remember it, let me just show. If this is the tRNA, it formed, but it just closed like this on itself, like this. So it forms what? Can you see that it is forming a paired ring and then a loop over here? Same thing happened over here. I hope this thing is understood now, what happened with it. So this clover leaf structure comes up because of what? Because of on itself, it changed its configuration and forms what? Certain double bonds. As it forms double bonds, okay, sorry, not the double bond, it forms the hydrogen bonds with complementary nitrogenous bases during or upon its own infolding up. Okay, now other way, in the 3D it is what, in 3D it is what, L-shaped. Okay, now the next thing is that it has what, it has the sequence 5' to 3'. Now on the 5' it will start with G nucleotide. Okay, whereas on this there are 3 nucleotide which is unpaired, that is what, ACC. Now this 3' end is called as what? It is called as acceptor end, because just think that ACC is free, means it can make a bond. Okay, so ACC is what? This 3' end is the acceptor end. Okay, what it will accept? It makes bond with amino acid. Just remember it, this will come up. Okay, so this 3' end which has ACC as their nucleotide sequence, which is unpaired, it makes this bond, whereas the other, this will make the bonds like this. There are these bonds present. These are what? The infolding hydrogen bonds. So these are what? Hydrogen bonds. Upon self-folding and complementary bases came in front of each other, it makes bond. So these are what? Hydrogen bonds. Now let's see over here. If you see this side beta, this arm is called as what? DHU loop. So this is what? This is a DHU loop. What is special about these loop? That it has 8 to 12 bases. Okay, so 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So it has 8 to 12 bases, which is not making any bond. That's why they are like this, a loop kind of thing present, which can't make bond. This site, it recognizes the amino acyl tRNA synthetase. Means this site will be recognizing that amino acyl tRNA synthetase. Okay, so it recognizes amino acyl tRNA synthetase. What it is, we will learn it. Let's come to this lower end, because as we are moving clockwise like this, so we reach to this point. This loop is called as anticodon loop. What do we call it? This is what? Anticodon loop. Anticodon loop means it's opposite to the codon, means it contains what? It might contains, the codon is what? 3 nitrogenous base together. Just the basic I have remember you. If you don't remember, in the next topic it will come up, don't worry. So this is anticodon, means it is opposite to the codons, the loop would be present. So anticodon loop, or it is called as recognition loop. Okay, which will joins with the codon thing. Okay, so it has what, around 7 bases present, forms bond with codon of mRNA, because anticodon opposite is there, and you know that whenever there is complementary thing present, it will join up. Okay, 2 and 3, that is 2. Okay, 7 bases are present on it at this anticodon loop. Now the last one over here, this is present, and this is called as what, T5C loop. Okay, so we call it what, T5C loop. T5C loop. Now this contains also what, 7 bases, 1 2 3 4 5 6 7. Okay, and it recognizes the larger subunit of ribosome. What it do, we will learn it. Process, we will learn it. So it do what, it recognizes larger subunit of ribosome. For now, just remember this fact. As we will do the process, you will understand everything. Well, that's all about transcription. I hope you would have understood this. And these are the different RNA that will be formed. And if the different RNA is being formed, means that by the process of transcription only. So we studied different types of RNA that is being formed by the process. We understood the process and the mechanism and the important concepts about it. If you like this video, give it a like, share it with your friends, because 12th half yearly exams are just approaching, so you can refer to this for sure in your genetics topics. If you like this video and you liking my video, don't forget to subscribe me. It motivates me a lot. Let me know in the comment section if you want certain changes or a better way of explaining this. I will try to do that as well. Well, on the reels and the shorts, we are also very active, gonna be very active soon. So just let me know in the comment sections what your take on it as well. Well, thank you.